food security. Dr. Erdogan, the floor is yours, and please proceed. And I'm going to stop sharing my screen here so that you can share with your screen.
All right. Thanks, Dr. Fairs. Hi, everybody. Can you hear me well and see the slides well?
Yes.
Excellent. Well, I'm so happy to be with you here.
If you, a suggestion, if you go to display settings and say duplicate, no, you can leave it there.
Okay, so let's see. I'm going
Put
to.
it, yeah.
Yeah, that's all right. Okay, great. So
you see the display settings here on the top? No. Keep it on presentation mode.
Okay, keep the presentation mode. Yes.
And on the top there is this. Yeah, click on that one. Choose the last one.
All right. How about that? Is this good now?
Yeah. Excellent.
All right. So, well, I'm happy to be with you all. Thanks, Dr. Fairs, for leading this excellent seminar series. And I'm also excited to be part of this community. Today, I'm going to share some of our research and link that with other opportunities in education, extension, and within this land grant mission. I'd like to talk a lot, but I'm going to keep it brief, and hopefully I will entertain some questions that you may have. First off, I appreciate Dr. Fair's introduction. I've been so fortunate to be trained in the finest institutions around the globe. A lot of great mentors I was able to work under, and it is my privilege and a joy to serve as the Executive Associate Director of the Cooperative Agricultural Research Center here at Prairie. It's an extraordinary opportunity to advance science, technology, and develop our students as well as serve our communities. So over the years, I had the understanding of the land grant mission, excellence in research, teaching, outreach, or extension. So as Dr. Fairs mentioned, this has been imprinted in my DNA, and I love it. All right, colleagues, there are a lot of grand challenges facing us. A couple of them I listed here, and those align really well with our college's strategic plans and actions that have been developed already. So among them, of course, is the food security as well as food safety. That is, how are we going to feed the world? And while we do that, how are we going to ensure the safety of the food, as there are a lot of foodborne illnesses? There is the connectedness between animal, human, and environmental health, i.e. One Health. They all depend on each other and interact with each other. We got to have the workforce ready future generation that we can develop through this land grant mission at this university. Things are changing, and as such, agricultural research needs to adapt along with the education of the frontiers who are going to shape our world. I'm talking about our students. In addition, we need to sort of translate the knowledge to extend to our communities to change lives through extension. What I'm talking about here, because of the dynamic changes, we got to adapt to those happenings, and not to mention the climate change, political problems around the world, as well as the changing landscape for agriculture and the consumer preferences. The good side, of course, is that there are a lot of opportunities, including the technological advancements, knowledge. So there are very few problems that appear to me not unsolvable. And usually those are wicked problems that there are not really good agreements on that. But most of the problems are solvable with the science and technology advancements. For example, with the robotics, unmanned aircraft systems, and then transferring the technology and the knowledge to our producers that would make a big positive impact. There are also some challenges, including public perception, the new generation lack of knowledge in agriculture. So those are some of the challenges, and I'm happy to share with you some of the publications here. I encourage you to read those. Of course, there are some new ones that really distill the grand challenges and some of the solutions. One Health is extremely important because of the soil chemistry and microbiome that are so important to support the plants that, of course, we use for food for humans and feed for animals. And guess what? Those animals also, most of them are, of course, livestock, are food sources for humans. That is, that the quality of the animal foods influence human health. So to me, that's a system science. If you think about this, CARC, our Cooperative Agricultural Research Center, with the natural environmental resources, social sciences, plant systems, animal systems, and all of that actually captured this concept really well. So I'm quite excited about how well prepared we are to challenge those challenging questions and find solutions. Another sort of introduction is that knowledge is the power indeed. Animals, including livestock, are critical elements of the ecosystem that we live in. The advancement in technology depends on the advances that we develop through science. As an example here, my graduate student, first graduate student, Dr. Osorio, and we were able to work with the community around the world to uncover the bovine genome. And that, of course, showed us that we are so close to cattle. You know, people sometimes raise their eyebrows, but turns out physiologically, genetically, we are very close. And we learn a lot from animal genome to advance biotechnology as well as impact other areas of science. So scientific development is critical for us to advance technology. Now, I would like to briefly share with you a research area that has been a passion of mine, which is to discover biomarkers to predict bull fertility or male fertility as it is applicable to other livestock species, and that to evaluate semen quality. I'll tell you the importance of this as I present over here within the time limit. Now, developing food animals with the economically important traits, i.e. phenotypes, is extremely important. This is because we got to have animals that are so efficient and systems that are sustainable and profitable for these production systems to thrive. Otherwise, they cannot survive in the changing landscape of the world economy.
Excuse me, Dr. Memili.
Yes.
Excuse me. Can you click on the display? Sorry to interrupt you, but I think we are seeing the previous slides. Yeah. Duplicate. Yeah.
How about this? Can you see it now?
Yes, great. That's it. Thank you.
Did you see my previous slides?
Yeah, I used to, but not now. So this is good now.
Oh, I see. So
yeah, we were seeing your next slide also preview. So, now okay. I see your slide and your face. So this is good.
I got it. Very good.
All right. Thank you, sir. Thank you. I'm sorry for interruption, but
thank you. So these economically important traits are important because without these traits, the farmers cannot produce milk, meat, or other products to provide to our communities. And some of these are listed here: reproduction, that is fertility, prolificity of the animals, the ability of the animals to withstand to challenges such as the climate change, changes in the sort of forage and landscape, and all of these things affect the animals, and they have to be well adaptable for those. Animals have to be healthy foremost in order for them to produce any food for us. For example, milk, meat production, these are directly influenced, of course, by animal health. As such, we have to ensure that animals are healthy. and that they have to grow sort of efficiently. We cannot these days ignore the cost of feeds, which is the most highest or most important cost of animal farming. As such, we need to have animals with efficient growth and development, and then the feed efficiency where they turn the grass into meat or milk. So how can we achieve this? Turns out these are quite possible with the advances of science. So I'm going to focus on the fertility and share with you some of our research in this area. So I would like to introduce you something over here, which is that the beginning of life, where over here you see the egg, oocyte, which is about 150 micrometers in diameter, one of the biggest, largest cell in the body, and then surrounded by lots of sperm. And so the fertility of the sperm I'm going to talk about today actually is defined as ability of the sperm to fertilize the egg, activate the egg, and then support early development. As you see, one sperm gets into the egg, and then egg seals the border. No more guys coming in here, because that would create genetic problems. After that, you start seeing the development of the organism. So this is the beginning, folks. This is the onset of life we are talking about, where embryo starts developing from 1 cell zygote to 2 cell, 4 cell, 8 cell. Of course, this sperm and the egg are haploid. And then when they unite at the fertilization event, then they, of course, each of these blastomers, we call them, are diploid cells. Isn't that amazing? Please say yes. This is a fascinating event. So I'm going to share with you today some of the research that my students and I have been doing to find out what makes the best sperm. That is, what are the markers and mechanisms that we can use to identify the best sperm. After all, a bull produces 4 billion of sperm in one ejaculate, and only one of those cells would be able to fertilize the egg. So how can we choose that cell to get the most out of the genetic and epigenetic contribution of the father? So why are we interested in the sperm? I'll explain you why. But before that, let me tell you briefly about a story of early life. At the beginning, as you saw, a sperm fertilizes the egg, and then it starts developing to become a zygote, 2 cell, 4 cell, 8 cell, and so on and so forth to become a blastocyst. Turns out this early life is extremely well regulated, as you can imagine, because there are a lot of molecular changes happening here while you saw those cellular changes. The importance of that is that if those events, the levels of genes expressed and the times they are expressed, if they go wrong, you cannot get that blastocyst in a quality way. As such, you can not expect a pregnancy of cow or goat or sheep after you transfer that embryo. Years ago, I had a very talented and motivated postdoc, Dr. Musir Lowell, who showed that during this transition of development from maternal to embryonic, there are a lot of changes that are genes expressed during these stages, especially at the 8 cell stage, the big burst of them related to chromatin remodeling and DNA methylation in these cells. That is, early life is developed both genetically, epigenetically, to give rise to the quality blastocyst that sets the stage for later development. So, what do sperm cells have to do with that? Well, that's what I'm going to talk about today in this short amount of time. How about a sperm tail? Please tune in. I will have some questions at the end, especially if you don't have questions for me, I will ask questions to you. First off, cell is extremely small, the paternal cell. Egg is big. Remember, it was 150 micrometers in diameter. Now, the cell is just, sperm is a couple of micrometers in diameter. However, our hypothesis has been that this small cell brings a lot of other things in addition to bringing half of the genome. Remember, that was the haploid male cell, this is the haploid female cell. They form the zygote diploid after fertilization. So this early development, as I mentioned, is critical for the quality attainment of the blastocyst, because if you have quality blastocyst, farmers transfer this cell into hormonally prepared recipient cow, and then 9 months later, a magic happens. So these are the connections between early life to blastocyst to live offspring on the ground. So my students and I, we've been fortunate to work with these cells, teamed up with a company in Wisconsin, Alta Genetics, and we found out that there are a lot of variations in the bull fertility. So this is the frequency, and then these are the levels of fertility. So that is deviation of fertility from the zero, either low fertility, minus direction, or high fertility in the positive direction. You see, there's a big difference between low fertile guys and high fertile guys. So you can imagine that the guys over here, unless they are extremely genetically valuable, would become what? Sure, soon they would become hamburgers, right? Because people don't want that, because you cannot get fertility animal on the ground, so you got to get rid of those animals right away. And so the question is, how can we determine the markers and mechanisms that could help us predict bull fertility, so that why don't we get all of the animals over here? And that has a huge impact, folks. Even a small percentage point increase in animal fertility, bull fertility, has a huge impact on herd fertility. The farms, some of them have 30,000 heads of cattle or thousands of animals in one farm. Do they have a lot of males? No. Why not? Well, first off, males, unfortunately, do not produce milk, right? And so farmers do not want males around, but they do need that to fertilize and for genetic purposes. So that's the value of our research, in that we got to have feasible mechanisms and markers to predict the phenotype, fertility phenotype. It's a phenotype that is influenced heavily by epigenetics, the changes in the cells I mentioned to you, chromatin remodeling, the DNA methylation, as well as heavily environmental influence this trait, because the inheritance of this trait is only 5 to 10%. That is about 90% or more factors, or the heritability of fertility depends on environmental factors. Therefore, the importance of nutrition, animal management, and all of that become very, very important, because using genetic tests like single nucleotide polymorphisms and all of that, we cannot predict the fertility well. In the economic side, this is a huge profit loss if we do not do a good job in predicting male fertility. As illustrated here, that really devastates producers and farmers, and of course, eventually affects the consumers. Another important part about bull fertility is that, as I mentioned, the impact of males. How can you have one bull having so much influence? Well, it's because of the assisted reproductive technologies, specifically here, sperm cryopreservation and artificial insemination. What is sperm cryopreservation all about? It is the freezing the cells. As I mentioned, the bull produces about 4 billion cells. So it's that process where you take the cells, cryopreserve them, and then ship them across the world. So you usually put about 10 million or so of these cells in each tube, freeze them, and keep them in liquid nitrogen and ship them around the world. And the producers then use artificial insemination to inseminate hormonally prepared cows so that one bull can essentially be used, the sperm from this guy can be used to inseminate millions of sperm. In fact, there are bulls with more than 2 million progenies. Isn't that amazing? So that is really the key point here, is that the male influence on herd production is very significant. However, these technologies, especially crop preservation, is not working well. As my former graduate student put it here, there are a lot of damage that occur as you put the cell, the sperm, into extremely cold temperature, negative 196 centigrade. As you put the cell there, you are expecting a lot from the cell to freeze, shrink, and then when you thaw during insemination, you expect the cell, this cell to be functional again. It is really fascinating. This is another research area that I hope to explore with our scientists at the International Growth Research Center here, and then hopefully we'll update you soon about this research. So in summary, we got to have the best sperm that is potentially fertile that we can use in artificial insemination. There are major challenges, however. For example, there are many bulls that have been genetically selected over the 100 years. However, they produce cells that are low fertile. Although they seem to produce tons of cells, their fertility is low. So why is that? My students and I, along with our collaborators, we have been testing the hypothesis that the reason these cells are low fertile is because of the molecular defects in the cells. That is, they have problems with the DNA, RNA, protein, metabolites, lipids that are part of the cell molecular anatomy. So our goal is, of course, very important for identifying specific new knowledge in the fundamental science of cell regulation. At the molecular level, what is happening with the functional genome, proteins, RNA, transcripts, and all that. So this research will uncover some of those molecular markers and mechanisms. Of course, the premise is that if you have the markers and mechanisms, you can then develop sort of precision agriculture breeding programs to improve genetic advancement of the herd. And you can use those markers to select animals or call the animals, get rid of the animals. So that is the rationale and significance of this research. We spent quite a lot of time in experimental design, where what is the irresistible idea to pursue? Where are the gaps in the knowledge base that link us with advancement in fundamental and applied science? So after we develop these questions, by the way, we visit with the statistician here a lot. So you got to have enough of statistical power, for example, to talk about research, well, experimental design. So that's very important there. So I'll briefly describe a sort of a general design of our approach or experiments. We have been receiving sperm from bulls with different field fertility, that is phenotypically distinct animals. And then my students receive these samples to do a lot of functional genomic studies, proteomics, RNA, metabolite, lipid, along with some of the flow cytometry, PCR, microscopy, and computational biology experiments, followed by in vitro fertilization to find the mechanisms and more insights. Why are we using these omics approaches? As you know, in the cell, we have the DNA transcribed into RNA, translated into protein, and those are functional genome that are very important for us to tell us what cell is going through. So what can we understand from these proteins about the cell, about the animal? That's the premise of the functional genome. Because it's the global proteome, RNA, all of these, they are very powerful because we have thousands of gene products. And in the past, of course, we had the gene chips. After Matrix started that, it has not changed. And now we have the next generation sequencing to get the blueprint of the DNA as well as the functional genome very efficiently, much less costly, of course, compared to the past. So let me share with you a couple of results that we have generated using these type of approaches. Using proteomics, this is the differential in-gel electrophoresis 2D Dodge, we were able to determine the proteome of the sperm from low fertile animals, from high fertile animals. And if you combine them and pseudo color that, isn't that cool here? This could be a great screenshot, screensaver here. You see the proteins differentially expressed. What do they mean? Well, if you look at the names of the proteins, they are very critical for cell physiology. And their changes are not extremely high. However, they have important functions in the motility. As you saw there, the sperm has to be very motile, forward-moving cell to be able to reach the egg and fertilize the egg. Molecular anatomy, these tubulins are extremely important for keeping the cells intact, protecting the cells from a lot of these free oxygen radicals. And therefore, we need a lot of antioxidants in the system. Of course, energy, sperm movement is powered by ATP. So we got to have enzymes that catalyze that reaction. And another area is the post-translational modifications of these proteins turn out to be very important. And some of our findings have turned out to be already actually confirmed by other scientists working with other species, including those studies in the mouse. For example, they found this ODF protein as well, we discovered, and this protein has a key role in sperm tail. This is the normal cell over here, and if you knock out this gene encoding for this protein, you essentially don't have a good sperm tail, and without this normal tail, the sperm cannot function. So it has been tested and proven that these markers are indeed important. So my student and my collaborators years ago, we published this study, and then we found 125 differentially expressed proteins between sperm from low and high fertility bulls. I'm happy to tell you that some of these proteins have already been tested and commercialized by others, and commercial companies have been formed using our markers. And people around the world have been contacting me and that they've been studying all 125 of them in their laboratories. So it's kind of good to see the impact of the study that my student has spearheaded at Mississippi State. We also looked at the epigenetic where, for example, in this study, sperm proteome, specifically the protein that's in the core of the chromatin in the cells, are very important because my student, graduate student Shula showed that this protein, which is like histone proteins in the sperm, are such that DNA is wrapped around them, and the tighter the DNA, the higher the cell compaction of that nucleus will be, and that will, of course, do two things. One is to protect the genome from damages, as well as help the sperm to move forward faster. So Shula showed that sperm from low fertility animals have a lot of DNA damage, and that they have lower levels of this protein 1. Remember, that's the protein in the core of the chromatin, the DNA, that helps the DNA to be much more compact in a very small size that cell cytoplasm has. So these low fertile bulls had sperm with lower expression of these, and then high fertiles had higher expression. As well as this location of this protein here is an image of immunocytochemistry, varied a lot. So the whole point of this slide here is that this image, there are a lot of heterogeneities in the sperm that you cannot just say the high level is better. It could be that high level is associated with abnormal cell. As such, we got to have integrated approaches with different experimental systems including cell biology, to interpret the results to generate new knowledge. My student from Brazil, Rodrigo, tested the hypothesis that there are a lot of histones in addition to great majority of the proteins I just told you that sperm actually contains. And he proved the fact that sperm actually, in addition to proteins, have a lot of histones. Those of you who study somatic cell biology, you know that histone 1, histone 2, 3, 4, linker histones, those are all very important to cell biology. And turns out there are some levels of these histones and that are retained in the cell. Of course, the higher the histone levels, the lower the fertility. Why? Because if you have a lot of histones, the fertility, the cell will be less compact. The sperm head will be very loose, cannot move forward, and it will be subject to DNA damage. I always would like to see, so that I could believe Rodrigo did both molecular approaches as well as the cell biology approaches to show that there are a lot of histones in sperm from bulls with different fertility. Earlier, my student tested the hypothesis that these cells have different RNA repertoire, that is the messenger RNA as well as the small non-coding RNAs. So sperm cell, because of the time, I didn't quite detail that aspect. Sperm cells are terminally differentiated haploid cells. We know that there are not many, there are not much activities as far as transcription and translation. So when we look at the somatic cell, the RNA would look like this, right? You have these ribosomal RNA nicely spaced out. When you look at the sperm RNA, you see this. So essentially, there's not much, you see, compared to this one to this one, you don't have much, except if you look at here, there's a thick band over here. What are those? Those are small non-coding RNAs, including microRNAs, piRNAs, and there are a few others coming up. So Nelida, my PhD student, showed that there are a lot of specific messenger RNA, although we cannot see them over here. In fact, this bioanalyzer imager looks similar if you put the water here. There are very small amounts of messenger RNAs that are very highly expressed in low fertility animals. Although we do not know the biology of these RNAs, that is, what functions they do, we know that these can be used as markers. You can get a pool of sperm ejaculate and aliquot them, test some of them for the expression of these RNAs. That could be very good set of markers. Later on, we did microRNA studies. Aruna showed that there are, in fact, distinct microRNAs that are associated with bull fertility. My student, originally from Tuskegee, came to research towards her dissertation in my laboratory and showed that there are specific enzymes, i.e. proteins, including sperm superoxide dismutase, very important enzyme associated with bull fertility. We expanded our research to seminal plasma together with the collaborators from Brazil. Arabella showed that there are specific protein markers as well in the seminal plasma. Remember, this is a fluid that comes with the sperm. This fluid is excreted from the extracellular vesicles there and the glands, and they are very important for sperm physiology as well as fertility as the sperm gets into the reproductive tract. So Arabella showed that there are a lot of proteins that could be potential markers. Erica, the postdoc from Brazil, showed that there are a lot of metabolites. Although much less as compared to the seminal fluid, there are a lot of metabolites that can be used in sperm to predict the phenotype, fertility phenotypes. More recently, Ali, the master's student, performed some studies on lipidomic biomarkers and showed that there are a set of lipids that are associated with the cryotolerance. As you remember, the ability of the sperm cell to be frozen and thawed and still alive viable, that's an amazing thing to expect from the cell. But there are markers, lipid markers, that we could use to evaluate semen quality and predict bull fertility. And one of my most recent PhD students, Nasir, showed that there are, in fact, epigenetic changes, acetylation or methylation of these histones. Remember, there were some histones in the sperm. The post-translational changes of these proteins actually modulate the sperm chromatin, making it either less or more compact. That affects the sperm function. So in closing, I would like to mention about the research conclusion-wise that the phenotype that we study is very important. We need to know what factors influence that phenotype. I illustrated here the fertility phenotype, which is very lowly inherited and extremely highly influenced by environmental factors, epigenetic factors or mechanisms. As such, doing functional genomics and uncovering potential markers is very important. However, there are also some genomic influence that we have to keep in mind, because when we select animals, we select for multi-traits, not just for one trait. So as such, we got to have the genomic markers, functional genomic markers, plus some of the other tests. For example, the motility, morphology of the cell, some of the molecular anatomy, integrity of the membrane. All these phospholipid bilayers can tell us a lot about the cell biology. And biochemical tests, what is happening in the cell? How much antioxidants or free oxygen radicals do we have the damage occurring from? And then more importantly, however, folks, I would like to mention that we got to have phenotypically distinct animals to uncover potential markers, and that in vitro studies are not as helpful, of course, and we got to do in vitro all of the functional genomic studies using samples from phenotypically distinct animals, and then develop those molecular tests that can be used as integrated approach. So finally, I would like to wrap up that this research is extremely important, and research in your laboratories as well, and those here in our college, because they provide experiential learning opportunities for students. This is the student that I would like us to develop, that is the confident, empowered with the knowledge and skills, because they are frontiers of agricultural scientists who will help shape the future of our world. And therefore, it's very important for us to link the research with education. Eventually, these folks will develop those skills, essential skills, along with the technical skills to have positive impact on science and society, not only in Texas and in the US, but in the world. There is also a very functional linkage between the research that we do and extension, because it is critical for our scientific community and students to realize translation of the knowledge. The new knowledge we have to translate to help solve the fundamental problems that the producers are facing. These are also important, the new knowledge, to develop new technologies. And as we get those biomarkers, we incorporate them to essentially selection and breeding of livestock. And that, of course, leads to precision agriculture. That's what we need. And then we advance the science, technology, produce food in an efficient, sustainable way to feed the world. And I mentioned the linkages between the soil science, soil microbiome, plant science, animal science, and human health. Very clear linkages there. So in closing, there are a lot of opportunities in the midst of a lot of changes, as Charles Darwin put over here. Ability to adapt, that is being agile, I think is the key. There are a lot of great happenings in the scientific front. We are fortunate to be in a country with well-funded environment right now for research And so I'm very excited, and I will be happy to answer your questions as well. Thank you.
Thank you, Dr. Erdogan, for a wonderful presentation. And the floor is open for questions and comments for Dr. Erdogan. And he covers a lot of ground.
Thank you all. This is actually, I don't know how I did this, but time-wise, this is pretty good because we have a few minutes for questions and any comments or recommendations as well.
Hi, Dr. Erdogan. I've got a couple of questions for you.
Go ahead.
You had the student that did the superoxide dismutase, the SOD, I assume it was SOD1, but when she was looking at that, did you see distinctly different ROS levels between the high fertility and low fertility?
Yeah. Good point. Thank you very much. At that time, we did not measure from that pool of the samples. But later on, a study that I did not present here, we did measure all these ROS levels and all that. They were quite different among the bulls. And I also would like to mention, and I appreciate your question, in that what we got to do is measure those ROS as soon as we collect them, so that the environmental impact as we collect the sperm are exposed to all of these oxygens would not interfere. So to answer your question specifically, we have not at that time when we did that study. Second is that a study of that kind is needed to really identify the endogenous levels as well as those that might be introduced in the exogenous as we collect the sperm. So that's an open research area right now.
Okay, my second question was one of the areas that I didn't really see covered in all of that, and I would like to get your input on the importance of it, is, you know, the glycocalyx of the sperm, and of course the sperm binds to the oviduct to hyperactivate and to prepare to go into fertilization. So there's a key role there with the lectin glycoprotein interaction. And have you looked at the differences in low fertility and high fertility bulls in terms of the glycocalyx of the sperm? And if not, how do you feel that would fit into the picture that you've just presented?
Yeah, we have not specifically studied that, those glycocactins and all this, but I know they're, of course, very important, as you mentioned. So our global proteome, one time I've seen the proteins being selected, but they were quite variable. And so I think that we got to study that. In fact, I think Dr. David Miller from Illinois somewhere, they've been studying those, and those are extremely important to look into that and see how much they are affecting ability of the sperm, especially after we put them into the reproductive tract. So again, that's another one that we have not studied at all. And however, they should be studied because, as you know really well, the sperm physiology is such that it has to do a lot of these things in different phases of reproductive biology. So I think that your question brings up an important one, in that we got to study a whole series of events during a whole different long periods of time. What I'm saying here is that just taking a picture, a snapshot, doesn't really tell us the whole story. So maybe during the time that we kind of looked at these cells, we were not able to detect those proteins, either because they were not expressed very well or our parameters were not designed to pick up those signals. But that's another area that I think it should be studied really well because it's a very important aspect of sperm physiology.
Thank you.
Yeah, thank you.
Great. Anybody else has a question?
Dr. Parks has promised a question to me.
Yeah, I do. Great presentation.
Thank you.
I'm not a livestock person, but you use the word economically important traits. So the economics part get my attention.
Yes.
Particularly when it comes to cattle, and I know we've looked at this, but My question really is this: what are the advantages or disadvantages of artificial insemination? And if artificial insemination is economically more important, why is it that so many producers are still using what I call the old-fashioned way of keeping the bull around? I mean, what is the advantage of even having a bull in the herd if you could artificial inseminate?
Sure. Thanks, Dr. Parks, for the question. That's very important indeed, because the artificial insemination, as you know, was actually pioneered here in the United States. I think it was over at Rutgers or somewhere, which is the ability to have one bull sperm to influence many animals. Like I mentioned, there are some bulls with progenies way over 2 million offspring, 2 million children from one guy. That's really amazing. So I'm mentioning that because it does have some disadvantages from genetic improvement point of view, or lowering the diversity, genetic diversity. Because in animal systems, we breed animals, and then you get the son of that bull, and then you use that to breed. It's not like humans, of course. So my point is, this can lead to inbreeding, and eventually we lose the sort of hybrid vigor. Mm-hmm. That's one major disadvantage that is actually quite difficult to recover from that depression, like inbreeding depression. We get a lot of genetic abnormalities eventually. So in fact, that's one of the major concerns in the dairy industry, where these animals have been extremely well selected because of the breeding. In fact, the United States has led the way to get these genomic tests. And even that has kind of enhanced that inbreeding potential dangerous. That's biological disadvantage. Economic point of view, the artificial insemination still has some disadvantages. For example, it's not well used in the beef industry. Why not? It's because of some lack of feasibility. You know, the beef herd, beef cows, these are almost like wild animals. You cannot maintain, you have to have systems to manage and do the artificial insemination, which requires docile animals, well-trained technicians, so you can do that. But in beef cattle production operations, this is not very feasible. And however, it's getting there. It's getting more and more kind of appeal to beef producers. So those are the situations with the cattle. With regard to small ruminants, I'm sure Dr. Foxford and Dr. Ho Watson would attest to this, that there are some challenges because of the smaller reproductive tracts in goats and sheep. It's difficult to do AI, artificial insemination, and that requires a technician, well-trained technician. In addition, the cryopreservation of the sperm is not well developed for these species, for goats and sheep and horses and pigs. There are still miles and miles of knowledge we got to generate. Although I think it's quite feasible with these technologies and scientific workforce that we have, but there's simply put gaps in the knowledge base and technology base that this technology is not well applied to other species. So as such, it's the combination of scientific disadvantage as well as some economical lack of feasibility that kind of is there in addition to great advantages that we've been enjoying. But I really appreciate this question. This is very insightful.
May I ask a question?
Yes, Dr. Kabroum, go ahead, please.
A few questions. The first question is,
the first one is free, but the additional, we're going to charge you for that.
Very
Go
good.
ahead.
I will. So is in animals fertility only about males? How is female infertility?
Okay,
And
that's very good.
Shall I
Yes, go
ask
ahead.
the second question? Okay, that's the low heritability of male fertility. So in
Yes.
one of the figures you showed above 0.5 and this. So if you select those above around 5, their offspring, they will not be like them, they will be different. And the third question is that about environmental effect. So what are the environments that favor fertility or
Excellent.
fertility?
Well, thank you so much. If Dr. Foxford, are you around? If you're around, Dr. Foxford, go ahead. Could you tell us a few things about female aspect of fertility?
Well, it's very based on the species also. Each of the species have their own unique issues. The goats have the problem with early regressing corpus luteum, which makes them lose their embryos. But one of the things that Dr. Memili mentioned in cattle, high fertility, the selection process using semen is used about 90+% in dairy cattle. But what that has led to is it's allowed them to increase the production so high that it's actually affected their reproduction. So we have to treat the females in cattle different and dairy cattle differently than we have to treat the beef cattle because we have affected their reproduction. We do the same thing in a lot of these species by selecting a given trait, and it downregulates their fertility. So there are a number of breeds in the goats that have the females have lower fertility than the other breeds. So, and that goes back to the whole inbreeding process. So we can use these technologies, but we have to use them wisely. As Dr. Erdogan mentioned, it needs to be this overall picture. But as far as the female fertility is concerned, it's not just the AI. There are other selection criteria, again, like with the dairy cattle for the production that have led to their decreased reproduction. And in each of these species, we have to determine what the problems are and figure out how to overcome them. And I go back to the dairy cattle because it's so researched now, but they use so much energy in that production that they affect the ability of their normal cycling, and it affects the ability of their eggs to mature properly and things like that. So again, it really depends on the species, but we can severely affect it through our selection processes.
Excellent. Thanks so much. As you see, actually, the answers kind of lead for us to think about the whole big system. There are so many things that affect the fertility, and which is kind of a segue to answering the second question. Is fertility transgenerationally heritable? At some level, yes. You see some of the research right now shows that there's some sort of transgenerational inheritance of fertility. However, because of the extremely high influence of the environmental factors, you bring the best fertile bull from, let's say, here to Africa, they wouldn't do well. Why? Because the environment is quite different. So as such, those other factors overpower the fertility phenotype. So because of that, the offspring of these animals, although they might be from high fertile bulls, are expected to be, are going to require extremely well sort of managed environmental controlled or farming strategies to be high fertile. So it's not a sort of a heritability, fertility wise to the next generation either. So that is still very low. That is, transgenerational inheritance of fertility is pretty low, just 5 to 10% at most. So, and of course, the bulls, interestingly, have a lot to do with their daughters' fertility. That's a very interesting aspect. I think that deserves some studies as well. So as far as the factors affecting fertility, heat stress is one of them. As the global warming and all those environmental impacts are coming out, they influence fertility very negatively in that increased temperature essentially inhibits the spermatogenesis. What we are talking about here is the population of stem cells in the testes are adversely impacted by heat stress. And the spermatogenesis, which is really a fascinating event, a lot of these morphological changes happen and sperm eventually form. Those are very negatively impacted because of the heat stress. The other one is the nutrition. We got to have the animals very well strategically fed. Of course, not to overfeed them, because if we have a lot of fat, let's say around scrotum, inside the scrotum that surrounds the testes, that animal is going to be low fertile. And therefore, the nutrition is very important. And I think that there are other factors, for example, that we still don't know what affects the animal's interest in reproduction, especially for those natural settings that Dr. Parks have mentioned. Most producers in the beef farming enterprises, they use natural breeding. They bring the bulls, but a lot of bulls these days don't seem to have any interest in breeding cows. So why is that? How can we increase their sort of desire? And that's another challenge that I think has a lot to do with the environmental impacts that needs to be studied. So other things, of course, are noise pollution. The all kinds of things. People have been doing research as to cow comfort. How about bull comfort? So how you manage the animals, reduce the stress, transportation stress, in addition to the heat stress and nutritional factors that influence fertility. These are very important. And I'm glad Dr. Foxworth mentioned a couple of other technologies. We can have all the technologies, but if we don't have these environmental things, we cannot really produce animals well. So there's a lot of wealth of things for us to do, and that what we know from the bull model can be applied to other models, including goats, sheep, horses, pigs, and many things. Actually, we can learn. One other thing I'd like to mention, that of course there's a lot of interest in cryopreservation because of the preservation of the gonads, the testes and ovarium. Because let's say people, especially young people having cancer, when they go through cancer treatment, the cells die. But the idea is that if we have methods to cryopreserve the ovarium or testes, we can then transplant it back, therefore sort of preserve the fertility. And of course, for humans, women would like to cryopreserve the eggs and do IVF later on, as well as for endangered species. So this technique of cryopreservation, AI, and other sort of the technologies that Dr. Foxworth has mentioned have a lot to do with other species as well. I just presented the bull model, which is one of the best studied and advanced model that I had a chance to learn in Wisconsin, which is one of the biggest dairy states in our country. But any other questions?
I think the time is past 12, past lunchtime. So I think this is a great presentation. I like the discussion. Although I don't want my colleagues to confuse AI for artificial intelligence for AI for artificial intelligence, which is another high topic these days. But I think this is a great discussion, and I appreciate it. I guess with that, I'd like to conclude, and we'll see you next week. So let's give our speaker a round of applause since he's for this great presentation today. Thank you again, and have a great rest of the day, and we'll see you next week.
Thank you all. I look forward to visiting with those of you who I have not yet had a chance to visit, but I'm so excited. Let us visit and explore. Thank you, and have a great one.
Thank you.
Thank you, Dr. Memili.